I'm going to talk mostly today about phylogenetics and some transcriptomic projects that we're doing in the lab. So as a person who does a lot of phylogenetics, I have to put the obligatory Darwin, I think, picture, but phylogenies have been updated significantly in the last few years, so we can do a lot of really interesting stuff, from epidemiological work with HIV, cancer, and Ebola, to kind of dinosaur genome size, which seems pretty crazy, and then most importantly, we can learn where beer strains come from. You can also get tattoos of these. I don't recommend them. There's a lot of uncertainty that you can't really represent in a tattoo. Nevertheless, in my lab, we do some empirical work and methodological work. The empirical work ranges from research on biodiversity and biogeography, and then some phylogenomics that I'm going to talk about, as well as developing methods to better construct phylogenies and study evolution on the phylogenies. I'm typically known for doing a lot of large trees, and I'm not really going to talk about those. So I'm not going to talk about this big tree of life that has the 3 million species that we published a few years ago, or this big sea plant phylogeny that is in test right now for that has 80,000 tips from GenBank or 350,000 tips, including the open tree of life and GenBank. So anyway, not going to talk about that. Instead, I'm going to talk about transcriptomes and how we can use them better for evolutionary ecology, especially for non-model organisms. So the data in GenBank, as a lot of us know, has increased dramatically. These would be the regular accessions to GenBank over the last, since 1985. If you add the whole genome sequences to that, it looks like this. So that blue line is the same line as it was before, and the whole genome sequence is added. These are enormous. So typically, in the last few years, what people have done is they've sequenced a lot of genomes and transcriptomes in order to do phylogenetic analyses. So this is kind of the impact it's had. These would be standard PCR-based analyses. You have one of a few genes. These are some really fancy studies that have, at least 17, 19 genes. And then people did all this sort of genomic sequencing, and then you ended up with 140 all the way to 8,000 with that Jarvis bird study. So this was the primary goal for a lot of that sequencing. I actually thought this is a really expensive way to make a phylogeny. So instead, I wanted to see if there were ways that we could use this data a little bit more effectively for asking questions about molecular evolution, more than just making a phylogeny. Not that that's not important, but I just wanted to explore this a little bit more for molecular evolution. So in order to do that, I needed to sequence my own data because these other sequencing projects weren't really generating the data that I needed. So I decided to do this on the Caryophyllales. This is a large clade of plants, over 12,000 species. The reason I chose it actually is because of the extreme disparity, though. So just to give you a taste, if you're not familiar with the group, they have most of the carnivorous plants that you're familiar with, the Venus flytraps and the sundews and all that. They also have plants that are adapted to really extremely cold environments. They have these cushion plants here, or Colabanthus, that actually, if you can see, goes all the way down to Antarctica. You get things in Svalbard and in the Arctic. But this also includes things that have photosynthetic adjustments like C4 and CAM. So this includes not only those really cold things, but also cacti. So you get really cold and really warm adapted things. And you also get these really crazy morphological modifications. So this is the Didieriaceae, which is a lot of fun to say. That's in Madagascar. Here are rock plants in South Africa. And then this is an epiphytic cactus ripsalis. So you get all this really cool ecological and morphological modification. It also isn't the case that all of these arose just one time on the tree. So this is a large tree of Caryophyllales with mean annual temperature maps, so the warmer colors are warmer temperatures. So this would be some expression of niche here, but you can see the adaptations on here. A lot of them arose multiple times. So succulents, for example, cold lineages, halophytes, gypsum adaptations. So these arose multiple times. So pretty good group to try and generate a bunch of data to ask some interesting questions. So what I'm talking about is based on the CNSF funded project with myself and Mike Moore as co-PIs, and then a bunch of folks that have worked in the lab or Sam Brockington, who was a postdoc at the time, but now is a curator at Cambridge. So we aim to sequence about 300 transcriptomes. We've done more than that now, to investigate phylogeny, molecular evolution, and the sort of climatic niche. The driving biological questions that we were asking were mostly things about life history and molecular evolution, the importance of gene and genome duplication, and specific gene family evolution. This last one I'm not going to talk about at all, but if you're interested in betalins or that kind of gene family diversification, we have some papers on that, including one that was just published this week. So let's just go through these. So I'm going to talk about the first two here a little bit more depth. So is there a molecular signature of life history across the genome? Well, in order to ask this question, we need to be able to use as much of the transcriptome or the genome as possible. So this was not possible a few years ago. To give you an example of why, this is a data set from the Vitaceae with Jun Wen and some colleagues. And they sequenced 16 species transcriptomes from these, and they found 400 orthologs with 0% missing data. So the transcriptome should have tens of thousands of homologs and orthologs, but they only could find 400 that basically they could identify between all the species. So why not more? Well, so this is a basic analysis that you would do for this. So you do some sequencing, and then you do this homology analysis where you're going to compare all the sequences, and then you cluster based on similarity, and you use these clusters. Some of them are too complex, and you filter them out, and some of them are nice and simple, and you use those for analysis. So the problem is you end up with a chart that looks something like this. This would be the genes sorted by how many species are in them. So if you had 35 or 36 species in an analysis and you did that analysis I just showed, you might end up with 1 or 2 genes that actually has all the species, and then most have many fewer. Okay, so some genes only have 4 species from this data set of 36. So you might expect it to look something more like this, where some genes are shared by more taxa, and then some genes might be clade specific, but something a lot more out here, where you might have tens of thousands of genes to work with. So this kind of analysis is very complicated. All I'm talking about at this point is basically this part, but there's all this stuff that happens in sequencing. So in my lab, we were trying to solve this particular problem, and so we developed a bunch of different methods that tackle this in different stages. I'm not going to go through all these, but if we wanted to go back to this typical analysis, what we basically did is changed this to look something more like this, where we actually incorporate phylogenetics in this analysis. So we take these messy clusters and we build phylogenies and we try and see if there are good genes in them. So this would be a messy cluster that in that Vitaceae thing would have been thrown away. We construct a tree from this, and you can kind of see visually that there are 2 clades in there. And then we clip this tree at this long branch, and we have 2 good genes now. So we don't need to go into the details here, but basically this 400 gene data set goes to 7,000 genes by doing this kind of simple analysis. And now we can use this data to ask our question. So our question was, well, so let me get this. So for the Caryophyllales, this data set looks something like this, where we had almost 100 taxa. Before we did this kind of tree-based analysis, we get the same curves that I showed you before for the Vitaceae, and we'd expect the blue lines, and what we actually get is something like this. So with 50% missing data, so not 0%, this is a larger data set, we have 10,000 genes that are useful here. So we have 10,000 genes now, as compared in the case of Caryophyllales to 300. So we can actually ask our biological questions. So you can also just build a tree. So this is a tree that we constructed that has almost 100 taxa, and it's using anywhere from 100 to 10,000 gene regions. And again, I wanted to ask a question about life history and molecular evolution. So the question is based on this paper that was published a few years ago that basically showed that molecular evolution is slower for tree species than herbaceous species. But this was all done mostly with chloroplast gene regions, and so it wasn't clear whether this is some sort of intron thing or whether it's in the entire genome or what this pattern looks like when you have more data. So if we ask this question with all of these genes, so we asked for 9,000 genes that we get from the Caryophyllales, we showed that more than 8,000 of them all show higher rates of molecular evolution for the herbaceous lineages in the coffin of that particular pattern that basically this is across the whole genome, this pattern that herbaceous species evolve faster than woody species, because we can show it not only in the chloroplast regions, but also the nuclear regions. So now with the last few minutes that I have, I want to give you another example of the kind of stuff that we can do with these data. So we're going to look at gene duplications across, gene and genome duplications across Caryophyllales and see what impact they have. So this is based on a number of studies that have been published in the last few years that have showed that genome duplications are correlated with diversification. So in this study by Tank, they had 9 genome duplications, and they showed that they were correlated with diversification. The way that you do this is you take a genome, you compare all the sequences to each other, and you're going to find a few gene duplications. But if there's a genome duplication, you're going to find a ton of similarities between this genome, and you're going to see this in some enormous peak and when you're comparing these genes on similarities. So you see these peaks over here. These are where we compare all the genes within this taxon, and we can show where there are genome duplications. So the question is, are these correlated with diversification? Well, if we do this on this data set of Caryophyllales, we found 13 genome duplications. That's as compared to across all angiosperms, where Tank found 9. So this is a significant increase when we increase sampling. We asked, if you continue to increase sampling, do you get more? And you do. So we added 50 more species, and you get 26 duplications. So the idea here is if you continue to increase, we'll probably get more. So Joe Walker in the lab, grad student in the lab, increased the sampling in the carnivorous group, and we found 7 just within the carnivorous group. So we're likely to find a ton more duplications as we increase the sampling, but again, we want to know whether they're correlated with diversification shifts. Well, now that we have 26 duplications in this tree, we are unlikely to see 26 diversification events that are increased rates of speciation that are correlated with that. Instead, I thought that probably genome duplications are more likely associated with shifts in adaptations, or as I might measure them in terms of shifts in climate because of the particular group, so movements to different climates. So to ask this question, I constructed a much larger phylogeny of the Caryophyllales, so over 5,000 taxa, onto which I wanted to map their kind of climatic niche. So I got a lot of geographic points. These are all the geographic points that I used to get climatic data. And then I mapped on the duplications we found from the transcriptomes onto this large phylogeny along with the climate to ask whether or not these duplication events are correlated with shifts in diversification or shifts in climate. So this is zoomed in, this is zoomed out. So you can see these various duplication events here, and then this particular climate data, and then the shifts in diversification, so the increased rates of speciation. So our expectation might be, if we're ancestrally in this particular climate space, in the duplication, we might see a shift, a movement, or an expansion, versus just staying the same. And we're going to look at plots that look kind of like this. So in this case, if we just pay attention to these two, these would be 2 different duplication events, and then this would be the climate. And what we see is in the duplicated lineage, an expansion in all these climatic variables. This is mean annual temperature, this is precipitation, and this is the general statistic that summarizes across them. And here's another group with a duplication, and we see the same pattern. So basically, when we see these duplication events, we tend to see this expansion. So just to wrap up, what we find with all of this sort of transcriptome data is that duplications, genome duplications, are not really associated with diversification. Duplications tend to occur before diversification, and instead we see these duplications associated with expanded climatic ranges. So the duplications, we might suggest, have something more to do with adaptation than with speciation, at least in terms of the processes involved. And that combining this sort of large tree paradigm with this transcriptomics is going to sort of help us understand a lot better a lot of these questions. Okay, so with that, I think I'm all out of time, but I just want to thank all the people on the project, including all the folks in my lab right now, some former postdocs, all that have a nice job, and funding sources, University of Michigan and the NSF. So, that.
Thank you, Steve.